Here we go. Good morning, good afternoon, everyone. Thank you for joining the IEEE EMBS webinar series on the frontiers of biomedical imaging and analysis. My name is Pingkun Yan. I'm an associate professor of biomedical engineering at Rensselaer Polytechnic Institute. I'm the vice chair of the IEEE BIP Technical Committee, which is the organization hosting the webinar series. So our guest speaker today is Professor Islem Rekik from Imperial College of London. Professor Rekik is a director of the Brain and Signal Research and Analysis, Basira Lab. Basira, I just learned this morning, means insights in its original form. So she is an associate professor at Imperial College London. Together with Basira members, she conducted more than 85 cutting-edge research projects focused on brain imaging and neuroscience. She is also a co-chair, organizer of more than 20 international first-class conferences, workshops, and competitions. So she's very productive. And in addition to more than 130 high-impact publications, she is a strong advocate of equity, inclusiveness, and diversity in research. So she actually is chairing several very important organizations, including the Women in the MICCAI WIM. She is currently the president. She also co-founded the International RISE network. RISE stands for Reinforce Inclusiveness and Diversity and Empower Minority Researchers in Low Middle Income countries. So in addition to her research, she is really contributing to the society with huge efforts there. So today, Professor Rekik will speak about her research on graph neural networks in network neuroscience. So without further ado, let's welcome our speaker.
Thank you very much, Pingkun. This was a very nice introduction. I'm very delighted to be with you today and take part in this IEEE webinar series. So today's talk will be about graph neural networks and their innovative application or extension to the field of network neuroscience, where we get to study brain networks or brain roadmaps. So let me start with a very simple hypothesis here that we have, especially in the world of neuroimaging and medical imaging in general. So generally, we hypothesize that the more modalities, the more data we have, the better the diagnosis. So that is an assumption that a lot of researchers make. And so that's why they aim to collect a lot of data, high quality data, with the hope of getting better diagnosis of particular diseases and disorders. So for example, here, one of the modalities that we could potentially use is like functional MRI, PET, CT, diffusion weighted imaging, and typically MRI. So however, if you guys look at the world, at this map of the world, this was published in the Lancet Oncology in 2021. And what you notice here is this is the number of MRI units per million inhabitants in each country. And in many of those countries, there is a very dire need of MR scanners. So half of the world here is colored in yellow, which means you have 0 to 2.4 MR scanners in those countries. So 0 means a few of these countries might not even have an MRI scanner at all. So the thing is, like, with having, designing methodologies that aim to collect a lot of data, it's very nice to have. However, this overlooks somehow half of the world. So what about them? What about us? So attention is very transformative in that sense that researchers need to put their attention where the need really exists. So there are different types of needs, and I agree, like there are when you have multimodal data, it's really cool to build these fancy models to make sense of that data. But in case you're not able to collect multimodal data, what are we going to do? Okay, so this is one of the questions that I want to raise. I'm not saying that I'm going to provide an answer to that during this talk, but it's just, a question that I would like you to think about. So there is a lot of attention that has been dedicated recently or pointing towards the this lack of imaging, medical imaging devices, particularly in low middle income countries. So, and this attention and, like conducted by different researchers across the globe can be transformative in the sense that we can design maybe novel methodologies or use AI to alleviate or to address this need somehow. Okay, maybe not directly, maybe indirectly. So you guys can see this is another. So all of these are published in really top journals: the Lancet Oncology, Clinical Medicine, Nuclear Medicine, Improving Women's Health in Low Middle Income Countries, the Needs of Diagnostic Imaging, Medical Imaging and Nuclear Medicine. Also, this covers this part. So these are very interesting papers that try to find a solution to this problem. So one potential solution, or the mainstream, I would say the mainstream solution to this problem, as published in these works and many other similar works, is to increase access to imaging facilities. What does that mean? It means that you try to enhance access to these imaging equipment. So you bring new MR scanners, you bring, you bring this technology to these countries somehow. You also need to train to build, capacity by training workforce, physicians, nurses who would be using these machines, train them to use, for example, digital software, etc. So this technology comes also with workforce capacity building. And this is with the hope to reduce inter-country inequities. So another thing is to remedy the infrastructural deficit. So trying to bring these, scanners to these countries. But as I said, many of these low middle income countries to rely on blind, which means they don't even have imaging and they go fully invasive, trying to understand, what is the disease that they're having without any imaging. And this is not something, it's really hard. It could be very difficult, particularly for practitioners in those countries, to do the diagnosis properly. So another thing, to increase the access, we need to provide e-training, to install imaging systems, and to have these e-trainings matching the level of expertise in high income countries. So these are basically mainstream solutions. However, they can be time consuming to expensive devices, scanners, etc. It's labor intensive. You need to train a lot of people, and it's costly to implement. So alternatively, we might want to think about how can we design more affordable, alternative, and faster solutions. And this is how AI can come to help. So today I'm just raising this question in general about imaging. So how can we diagnose early and better with minimal resources or with minimal limited data and at lower costs? So this is the most important question that we'll try to partially answer, but not fully answer. And I will explain why it's still a new research field. And I do believe there is a whole lot to do within this realm. So first, let's start with our nice variable X. So we do have here our data. So this is, the data set that we have collected. Hypothetically, we hypothesize that the data is affordable to acquire. It's not that expensive. It's accessible. It may have low to middle quality. So that's not a problem if it has low to middle quality. And then the goal is to predict this X hat, or to generate this X hat. That is the data that is costly to acquire, inaccessible in many countries, and of high quality. So basically what we want to do, we want to use AI or, machine learning, deep learning models that we will design and innovate in order to map this low quality data to the data that we want to have. So basically, if a particular hospital in the world has only limited resources, but here when we say limited, we still need to start from a particular imaging modality. We cannot start from zero. So for example, CT or ultrasound might be more affordable than MRI in particular countries. Okay, so we can start from there and then try to predict the costly, high quality data that we don't have the machine to acquire it or to kind of, like to make the scan, okay, of that particular modality, like MRI. So getting MRI, for example, from CT. So here we're talking about imaging, okay? But in this particular talk, I'm focusing on brain networks. So generally, let's take one step back and think about or remember, if you guys know, I'm not sure if you're familiar about brain connectomes or brain connectivity maps. We call them connectomes, connectivity maps, brain network, brain graph. It's the same thing. So basically it models the relationship between different parts, anatomical parts of your brain, okay? So typically we use what we call MRI. Conventionally, the only modality that has been used, to the best of my knowledge, is MR scanner to magnetic resonance imaging to create the brain graph. Okay, so here you can see different nodes connecting different parts of the brain. So the one that was the sequence that is the modality that is typically or conventionally used is actually functional MRI and diffusion MRI. So these two modalities are not conventionally acquired in many clinical facilities all over the world, and they take more time to acquire. So it would be interesting to kind of find a more affordable way of creating brain networks without resorting to acquire those sequences of functional and diffusion MRIs. Okay, so like lowering the time, the acquisition time, the cost, and trying to build brain networks from only T1 weighted MRI, which is the basic acquisition sequence. Okay, so here we introduce what we call a morphological brain network. A morphological brain network is a connectome that models the morphological similarity or dissimilarity between different regions, like in terms of geography or morphology. How is this brain region similar to another one? Okay, so this work was, I'll give you just a brief overview about how we do this in a second, but let me explain. So let me start with this. Sorry. So we're starting with an affordable, relatively affordable morphological brain network that can be acquired from the basic MR sequence without resorting to other sequences. And what we want to do is to learn how to predict other types of brain networks that might be costly and of high resolution, for example, across different domains. So here you go from the morphological domain and you map into diffusion, connectivity, into functional connectivity, which models the correlation between neural signals in the brain. And the second axis is actually resolution. So we go from a low resolution brain where we have few nodes and we try to create, to predict and generate X-hat brains, like different types of brains that are at high resolutions, which means we have more nodes. The third axis is actually time, which means with a single acquisition, without resorting to acquiring more scans of the patient, we don't need to invite them to have more scans. So you're lowering also time and cost. You're able to predict how this, particular brain network will evolve over time, whether it's a typical evolution or atypical, which means healthy or disordered, for example. Okay, so we have developed more than 60 works and to design this, what we call predictive learning in network neuroscience. And this is, predicting brain networks across different domains, time, and resolution. So how do we derive actually a morphological brain network from T1 weighted MRI? So if you guys are interested, you can have a look at these videos. Like this could be a whole talk about how we do that. So if you go to the Basira Lab YouTube channel, you'll find these first publications back in 2018, and we explain how we derive the morphological networks from T1 weighted MRI. So moving up next, so here, as you guys know this in this diagram, what do we see in terms of the distribution of the publications in the field of medical image analysis? So this was a paper that was published back in 2018 in Nature Communications, and you can see that the majority of the papers they focus on primarily segmentation. You have classification, also disease probably, or detection, object detection, tracking, retrieval, reconstruction, but 2% is at the time, a few years ago, was focusing on prediction. And this was one of the motivations that pushed us into kind of exploring this direction. So let's talk a little bit about this predictive learning or predictive intelligence across different dimensions. The three axes I've talked about. So first, we have what we call the single graph prediction across domain. So given a particular brain graph, here you have, for example, 4 different regions in the brain. So this is a very, by the way, this is a generic abstract framework, which means you can apply it to other types of networks, not only brain networks. So our application area is brain networks, but these models that I will present today are very generic and can be applied to similar data sets. So given a source graph, we aim to learn a mapping or build a network that is able, a neural network or deep learning model, to predict a graph in a completely different domain. Like, for example, here we learn how to infer or predict functional connectivity from morphological one. And these are completely different domains. They give you different information about the brain and how it works. So for the multi-graph prediction, we aim to solve even a more challenging problem, which is, multi-graph prediction. So you're going from a single source graph, like morphological, for example, network, and you're trying to predict a set of graphs right there. So here, target graphs across different domains. You have n domains here with different types of edge connectivity. So this is what we call multi-graph prediction. So the second axis is the resolution. So to the best of our knowledge, we were the first to introduce the concept of graph super resolution. So in the literature, you have lots of works on super resolving images, trying to create higher resolution, images from low resolution images like 70 like MR, from 3T MR or 1.5 Tesla MRI. You have also super resolution computer vision. In graph theory, when we talk about graphs, we try to filter them, compress them, to downsize them, but there isn't the concept of creating a bigger graph from a smaller one. And this is where we introduce the concept of a graph super resolution. So you go from, given a low resolution graph where you take a brain, you parcellate it into different regions, like big blobs, for example. It takes, let's say, 1 hour to create this from the image itself. Then you have a higher resolution graph with a more refined parcellation, and with this refined parcellation will take a lot of time, like hours, let's say, to transform the image into this high resolution graph. Okay, so for us, instead of like trying to to circumvent the need of costly image processing, we directly learn how to predict the high resolution graph from the low resolution one. Okay, and that's what we call super resolution, graph super resolution. Now for the third axis, the prediction here is a long time. So given a baseline brain graph, we aim to predict its learn a mapping that is time dependent, f of t or ft, that is able to predict the evolution and the changes in the brain connectome over time. So these are like basically the 3 different axes. Now there is one fourth, I would say, component that might permeate or help the learning or guide the learning along these 3 axes, which is what we call brain templates or template-based learning that we also introduce. So the graph integration here, there are lots of works on graph fusion, graph integration, but what we have introduced here is the concept of multiple graph integration to create what we call a single brain template of a population. So you have a healthy population, healthy brain connectomes. You learn how to merge all of them into a single matrix, a single graph, and you have another population with atypical disorder, and then you merge all of them and you'll have these 2 templates. And by comparing these 2 graphs, you can easily spot or identify the differences between a healthy population and a typical one just by simply looking at this fingerprint. So what is this basically? Each subject here is represented as a row. So you have a row of you have a multi-graph here. It's like it's an expanded tensor or flattened tensor. So you go from view 1, subject 1, view 2, view 3, etc. So each subject is represented by multiple networks, not a single network. Yeah. And then you have multiple subjects, and the goal is to learn how to integrate them while taking into account the heterogeneity and the multiple domains that they come from or that they are sampled from. And these domains are actually the different views. So now using this template-based learning, we can use it for graph super resolution learning, multi-domain, cross-domain prediction, and also time evolution. So you can always use templates to improve the quality of your training of your network. So once we predict the data, then what do we do? So you might ask me, oh, you're just predicting all these networks. So what then? What do we do? So ideally, we would use the predicted data along with the ground truth one, and then we will train a classifier to enable us to identify biomarkers. Also, like do some disease classification here, like learning how to disentangle or distinguish between disordered subjects and healthy subjects. So this is the goal, is to do somehow like a cross-modality, affordable data generation that enables you to boost diagnosis and to understand how the disease affects the brain, for example, in a more holistic way. So all these works are actually on YouTube. So like we had lots of works along these directions. So I'm going to just I've sampled only three examples because we have just 1 hour to cover today. So if you are interested in learning about this, you can check. We have like more than 39 oral presentations and journal presentations on this channel here, and like these are lengthy, detailed videos. Today, I'm just going to give you an overview. If you're interested also in running the codes, you can see how to run the code by checking, the code videos. And you have also access to the codes on the Bocera Lab GitHub. So we try to support open science along multiple directions. So these works also have recently been published in as a TPMI review paper covering GNN models and network neuroscience from 2020 to 2021. So if you're interested, feel free to read the paper and check out also the GitHub repo associated with the paper. So let's start with the three examples. And the first one is the cross-domain prediction. So here, for the cross-domain prediction, this was a work presented by my former PhD student and accepted at a MICCAI 2020 conference and then extended into a medical image analysis journal. So let me explain briefly what is this, what's the aim of the cross-domain graph prediction. So the problem that we want to solve here is how can we design or like a graph neural network or geometric deep learning framework that enables the prediction of multiple graphs, target graphs across multiple domains from a single source graph. So this is the question that we want to answer. How can we do this multi-graph generation model? So at the time, the existing works were primarily focusing on a one-to-one mapping, which means you predict a single target graph from a single source graph. So no, there weren't any works that tried to solve this problem jointly, which means multiple from one-to-many mapping. And the second limitation is like the existing works, they overlooked the local topology preservation. So we know a graph has a topology. For example, what is topology here? We know that this node has two connections, okay? So that's the degree of the node. The degree of a node is a topological feature of a node in a graph. So there are lots of topological features like centrality and like hubness of a particular graph that we need to preserve when we predict, which means when we have the target graph and we compare it with when we have the ground truth graph, brain connectome, and we compare it with the predicted one, we need to have similarity in topologies, not only in adjacency matrices or edge weights. So what we proposed here is to use a graph, a convolutional neural networks. So basically in this problem formalization, first, we have a set of source graphs. So for n subjects, S1 to Sn. And this is for training. So we have also a set of target graphs across m domains. So for each population here, and these are of course the same subjects, okay? So we have we construct what we call a feature matrix. So for each node, we extract a feature vector that depends on its connectivity. So we have all these subjects represented by features. And then we define also what we call a similarity, a subject to subject similarity matrix that will enable us to, like create a graph of the population in the source domain. And we do this basically for every single domain, the source one, target domain one, target domain M, etc. So then we use what we call an encoder. We design again a multigraph. So this is a multigraph GAN network. So in this GAN, we have an encoder of the source domain. So we try to encode, learn an embedding for every single subject in this graph, and this is how we get the embeddings of all subjects set right here. So these are the embeddings of the subjects that we learned using the similarity feature, the similarity matrix between subjects and the original features. So once you get, these embeddings, so then if you guys are familiar with GANs, there is an issue what we call the mode collapse. So if you have if you have heterogeneous data with a heterogeneous distribution, so let's say your distribution, the source domain is heterogeneous, then you might, if you want to generate your data across different domains, you might collapse in a single mode. So which means it captures only one single peak of your distribution, one single mode of your distribution. So to kind of circumvent this problem empirically, we didn't provide any proofs or demonstrate that, but there should be some theoretical. It would be exciting to develop some theoretical works or proofs around along these lines. So basically, we took the embeddings for all these subjects and we clustered the subject based on their learned or encoded embeddings into different clusters. So we disentangled the heterogeneity, and then now we said we're going to design or to train a decoder for each cluster individually. So we had a decoder network here, G of our GAN, to generate the output, to predict the output, feature embeddings or the features for the subjects in the target domain T1 using different clusters. So we have cluster 1 and cluster C, and then for the other domain, for domain TM here, we have also cluster 1 and cluster C. So for each cluster, we do this training, and we predict a target graph. So all these decoders are regularized by a single discriminator network. And then basically when we want to do a prediction, we will use a testing graph that will be fed into an encoding network and decoded through all these clusters, like for example, cluster 1 to C for domain 1. And then we simply average the predictions to get the final predicted network in the domain 1. So this is how it actually works. And we added some topological loss here. So this topological loss allows us to kind of, preserve the topology of the predicted graphs in the target domain compared to the ground truth one. So we enforce that during training. So in our results, we use 310 subjects from the IPMI data set, and we have three types of networks. So quickly, in the MICCAI paper, we used a single random split, 90% for training and 10% for testing. But in the media paper here, the journal, we used cross-validation, and we evaluated multiple times and repeated the experiments. So as you guys can see, in these are the comparison methods. So there weren't any methods that solved this problem before. So we took MWGAN, which was designed for images, and adapted it to graphs and evaluated it. So you can see this is the Pearson correlation. So the higher, the better. These are the MAE errors. So the lower, the better. And you have MAE in betweenness centrality, closeness centrality, and eigenvector centrality, different topological measures. And you can see that the multigraph GAN, when it was enforced or, by a topological loss using betweenness centrality and eigenvector centrality, we had the first best and second best performance compared to these, modified methods. So if you're interested into learning more about this and in detail with lots of, with all the maths and all the detailed formalization, you can check out the 20-minute video on YouTube, and the code is also available. So now let's quickly move to the second example. which is the cross resolution. So from cross domain to cross resolution. So in this work, this was a paper accepted at IPMI 2021 as oral presentation, also presented by my former PhD student Islem here. So what is the problem that we want to solve here? So as I mentioned earlier, so what we want is to learn how to predict intermodality non-isomorphic brain graphs from a base graph. So non-isomorphic, one of the ways to break isomorphisms between graphs is to change the number of nodes. So that's very kind of obvious. So here, conventionally, the existing methods that try to solve, well, there isn't any method that tried to solve this problem, but the methods that existed back in 2021, they only worked on predicting graphs with similar number of nodes. So going from NR by NR nodes to NR by NR adjacency matrices, okay? So also the existing works did not take into account the gap or the domain fracture in distribution between the source and the target spaces. So you have a discrepancy here that you can see between, for example, morphological brain connectivity and functional brain connectivity. The distribution is not the same. And what we want to learn is a mapping that allows us to predict, you know, these graphs, the purple ones from the pink ones. So what we propose here is to solve both problems simultaneously, which means we super resolve the graph. So we predict a higher resolution graph, NR prime by NR prime from NR by NR. You can see the matrices are larger. And we also learn how to align the distribution of these networks using, you know, the KL divergence laws towards the distribution of the target network. So we design or introduce an alignment network that enables to better predict the target super resolved graphs. So let me explain how the proposed architecture Iman GraphNet works. So first, we have a block that we name an intermodality domain aligner. So the idea, guys, is actually simple. It's not too difficult, but it was very effective. It gave really very good results. So here, first we have the X1 to XN training data, neural graphs, neural subjects. So we have X training graphs or connectomes, brain connectomes for the source domain. S is the source domain. So we define what we call a graph aligner network here. So it's composed with different layers. So you have graph convolution, dropout, and batch normalization. And then through this, so let me just hide this before, sorry. So, and then through this aligner network, what we want to minimize the KL divergence over a long training between the source distribution, which is the purple one, and the target distribution, which is the blue one. So you can see here, they're completely aligned. So after basically these networks pass through the aligner, their distribution will be aligned to the target domain. So you still have X source network, but it's aligned arrow towards target, okay? So in distribution. So once you have these source networks aligned in distribution to the target domain, then we design or we propose a non-isomorphic intermodality graph GAN. So basically here we do have we take the aligned networks, source to target, and then we learn using simple, you know, linear algebra operations that we introduce through the network, how to change the resolution of these graphs towards the high resolution that we aim for, which is NR prime by NR prime. And then that's how we get our X1T target hat, which is predicted network super resolved brain graph in the target domain. And these are regularized by this block is regularized by a discriminator network that discriminates between the predicted target super resolved graphs and the ground truth one at high resolution. We always have the ground truth data. So using this Iman GraphNet, we actually had, we used the SLIM data set. We evaluated it on the SLIM data set. So we had 150 morphological connectomes at resolution 35 by 35, looking at the cortical regions only, and then a whole brain, functional connectomes at resolution 160 by 160. And I'm going to present here the results. So here we had different ablated versions and different ways of doing alignment. So first we have Iman GraphNet without any alignment, and you guys can see we have the MAE error here, which is the distance between the the MAE error between the ground truth graph adjacency matrix and the predicted one. This is MAE in closeness centrality, topological measure, MRE in betweenness centrality, another topological measure, and eigenvector centrality. And you can see that without any alignment, we had the largest error. Now, we use the statistical alignment here, which means we replace the whole learning base end-to-end block, because these two blocks communicate. They're learning co-trained together. So we removed that previous A block aligner network and we replace it with a pre-statistical alignment that you can do just like by aligning the data manually. And so manually means like using just simple without any learning. And then you see here the error with some statistical alignment, we have some improvement, decrease in the error. Now, we also introduced what we call a Pearson correlation loss function. So when we remove this loss, still we have like some alignment. There was a decrease in the error. Now, Iman GraphNet without topological loss still decreased, but when we use Iman GraphNet with the topological loss PCC and the alignment, you see you have the best results, the lowest error here. Using a two-tailed pair t-test, we got the best. Also, we got a p-value lower than 0.005. So here we did also clinical inspection, which means we looked at ground truth and predicted networks top connectivities, and they were very similar. So there was a large overlap between the predicted top connectivities of the ground truth super resolved, super resolution graphs, functional connectomes, and the one that we predicted. So that was a qualitative also analysis. So this also work is available on GitHub and also on the YouTube. You can watch the video. So let's move to the next one, which is cross-time prediction. This is the last example. So here, this was a recent work accepted in a Prime MICCAI workshop 2022 and published recently last month in IEEE TMI. So what is it that we try to solve here? So this is a very actually exciting problem. I don't think there's a lot of research, and if you guys find anything around these lines, please feel free to drop me an email. I'm very interested. I don't want to miss other people's works. So what we try to solve here is like to design a federated learning model that learns from incomplete data sets or temporally varying time series data sets, which means you have multiple different time points that do not correspond across your clients or hospitals, and you still want to learn how to predict the evolution from a single observation. So basically, the problem goes like this. So given to learn how to predict this over time, but in a federated manner, while basically looking at the problem of missing data points. Okay, so here, and just in a nutshell, I'm going to give you the idea. So first, we have what we design a GNN-based time-dependent prediction model. So this matrix is so important. So you have hospitals here, and you have time points. So some hospitals have many acquisitions. So one, it means acquired, available. And for when you have 0, it means it was not acquired. So the best scenario when you learn is when you have 1, which means you can have, for example, train a symbol GAN to generate this next time point from the previous one. Okay, or the current one. So this case is the simplest. That's why here we train a generator GNN. Now, when you do have, for example, the second case, so we have 4 cases here. The second case is when you have an existing time point, the next one is missing. How are you going to train this hospital, the model for this hospital locally? So what we did a self-encoder. So you self-encode, which means you just pass on almost identity map here when you train it locally before you get the information from the server in the federation round. So this one also, you can, because you have the next time point, so you can learn how to generate from this one. So hypothetically, this time point has had is this case, which means you self-mapped and then you kind of learn how to generate the next one. And the worst scenario is basically when you also have consecutively missing time points. So some hospitals might have only, imagine one hospital acquiring one time point, another hospital acquiring, 5 or 6 time points, right? So this hospital is not privileged at all. So in a federation process, it can exchange its model or learn from the average models of the other hospitals and boost its, its predictive power through this federation process, and learn how to predict without even having data to train on, which for me is really fascinating, because you don't even have data and you still learn how to train your model to predict something, right? So so these are the 4 different cases, and here we proposed a mixed federation strategy. So the first one is simple. So these are in the state of the art. This wasn't the biggest innovation in the work. So, but one of the components that we proposed. So first, you have a GNN and wise weight aggregation, which is simply averaging. This is Fed average that was first introduced, I guess, back in 2014. And then you have pairwise GN and layer weight exchange. So we alternate between these, the exchanging models and averaging models. And what we have introduced in the TMI paper is an ordered way of training hospitals. So basically, these models should not be exchanged randomly. So the hospitals with more information need to help the hospitals with less information. And we presented a way of ordering this model exchange and federation in an autonomous and automated manner. And this was one of the key components that we also introduced. So if you're interested, have a look at the video and also the paper. So here, the data set, we use 113 samples from the OASIS data set. Each sample had 2 observations, 2 time points. We had lots of also simulated data in the TMI paper. So we simulated sequences with 6 time points or even more, because we didn't have real data like that. So we wanted to kind of draw from multiple distributions and show that this model still works when you have dense acquisitions. And that's actually when it gives lots of good results. So here we compared against a 4D GNN, which is just, graph neural network that predicts without any federation, the 4-dimensional evolution of the baseline graph. And then we had a 4D fed GNN, which is just simple averaging. So we use the layer-wise aggregation. And you can see in these results, across 4 hospitals, we have 4 hospitals. So without any federation for time point T1, the MAE error is really high. Now, when you federate, it really drops, which means generally federation really helps. So for the 4D fed GNN plus, we had the federation and the model exchange with different these case scenarios, and you guys can see that we had a further improvement. And we added the 4D fed GNN plus plus with the ordering sequence, and we had even another improvement. So that was the TMI paper. So T2 time points at the second time point for the real data set, you guys can see also a very nice decreasing trend, reproducible using this federated learning strategy. So the paper is also available, and feel free to watch the video. Also, there is a video. And yeah, so if you're interested in the data set is actually online. So we have shared it. We shared the morphological brain networks with 2 time points. So there was a paper that we published, I think in 2021. It was a competition on Kaggle on how to use simple machine learning methods to predict the evolution of brain connectivity from a single time point. So there were 20 competing teams, and in the paper we compare all the methods, and we explain why we draw different, we have different analysis figures of their performances using different evaluation measures. And you can see that the first team who ranked up, like, you can look at their methodologies if you're interested, and the paper is also available. So I'm not sure we have time to cover this, but I'm just going to go over maybe 3 more minutes and then take a few questions if you guys have questions before we wrap up. So here, if you guys remember, I talked about creating a CBT, a template network, a fingerprint network from a set of multigraphs. So what is a multigraph here? So if you guys can see, we have each node, 2 nodes are connected not only by a single edge, by multiple sets of edges. So this is what we call a multigraph. So the nodes are the same, but the edges or the edge types are different. So in this example, we want to learn how to integrate these multigraphs into a single connectional brain template and use this template to for learning or downstream learning tasks in brain connectivity, regression, and classification. So this work also got an early accept in MICCAI 2020. Was presented by my undergrad student Burak. So just quickly going over this. So the goal is to learn how to integrate these tensors into a single connectional brain template. And he proposed the first graph integration network that enables to merge or collapse all of these multi-tensors into a single matrix. And why this is interesting, the work is actually very interesting in the sense that we introduced a new loss function where we compute the distance between this CBT, the center, to all the views of these tensor. We try to minimize the distance between the CBT here to all these views, right? But then instead of using all the training set, we randomly selected the subsample, and we showed with this random sample selection, you optimize, you have like a faster training, but you also a good representativeness of your training cohort through this random subset, training subset selection. And you can think about it as a centroid discovery or learning. And you might think about using an average, but averaging doesn't give you good results because there are lots of nonlinearity, and also you might have some outliers in the data, so that doesn't give you a good center. So so we did this in a fully learnable way using graph neural networks, and we showed that our paper has outperformed conventional or a state-of-the-art method, a Nature Methods paper published in 2014 called a similarity network fusion method. We also compared it against other types of integration methods, and this geometric deep learning or graph neural network was really powerful. It reproduced over and over again across, I think, 8 data sets, better results across different evaluation metrics. So why this is important? You might ask me, oh, okay, we discovered the center, then what do we actually use it for? So basically what we did, we showed that once you have this center, so this matrix, okay, of a population with Alzheimer's disease, and you have another population with late mild cognitive impairment, you, by simply calculating the absolute difference between these two matrices and taking the highest values, you're able to spot the brain connectivities that change, that mark the difference between these two brain states. And then what we did, we took an independent learner, an SVM, a multi-kernel learning classifier that takes, that selects features, and we found about 70% reproducibility between the markers that were identified using a simple CBT matrix differences versus a heavy learning-based approach to discover the biomarkers using all training samples and all multigraphs. Yeah. So basically, this is very helpful in terms of population fingerprinting, comparing different brain states across multiple cohorts in a very simple operation that we can use, which is subtracting two matrices. So if you're interested, you can read also the medical image analysis paper. It's long, so we extended this also into MGNet, multi-graph normalizer network, that enables you to normalize all these networks and integrate them into a single template. So I'm going to skip reproducibility. It's a very interesting topic, but not very much explored, I guess, in the literature, but I'm going to skip that and talk in the last 2 minutes about the RISE network. So so guys, I don't know if you heard about the MICCAI conference thing, a few of you or many of you might be familiar with that. So if you are, we have a few years ago, 2 years ago, we started what we call the Reinforced Inclusiveness and Diversity Network, RISE MICCAI, that enables to provide free training to students all over the world. We through this, we want to empower, we want to have capacity building. So if you're interested, please scan this. Feel free to join us. You can read about, you can just Google RISE MICCAI. You will find the web page. So the RISE mission is to basically propel, as I mentioned, inclusiveness and geographic diversity, because we have seen that there is a lack of participation from 104 countries in the world to one of the top conferences in medical imaging. So we want to have more young researchers coming and rising from those countries, joining our network, doing excellent research works along those directions, and along, like in terms of AI and medical imaging, global healthcare. So we want to strengthen the presence of minority researchers, empower biomedical image analysis researchers, pave the way also for future stars to rise from those communities, build connections, strong networks at a global level. And yeah, so I always say we never rise alone, we always rise together. So if you're interested, please, feel free to join us. We have lots of upcoming events. So we're going to organize a bi-weekly journal club that is on Zoom. We're going to give you a paper 1 week beforehand. You'll read the paper, come and join us to discuss the paper, learn about recent trends in AI, medical imaging, image analysis, data analysis in general. So hopefully in the future, we'll also organize MICCAI challenges, hackathons. The last November, we organized the first RISE MICCAI winter school in collaboration with the Africa Network. So it was also for 3 days and online. We have top speakers joining us. So yeah, so we'll have lots of events, summer, winter school tutorials. If you're interested, have a look at the website, fill out the form, you'll be added to the mailing list. And if there is something up that will be advertised soon, you should be able to learn about that. So yeah, so I would like to thank all my students across all the years. I started my research lab back in 2017, so I don't have all the pictures, but this was a recent hangout prior to MICCAI. Yeah, so I'd like to thank all my students, and I thank you all for listening, and I'm happy to take your questions.
Well, thank you so much, Islem, for the wonderful talk. It's very, very impressive. Yeah, tons of
Thank you.
high quality works, and I also appreciate that basically all the your papers come with codes. You have the GitHub links there. That's terrific for reproducibility. That's what we want to see for the field. Wonderful, thank you.
Thank
we
you.
already got quite a few questions in the chat window. I'm not sure we have time to finish all of them, because I know you have another meeting at 10. But we may actually try to answer the first several. I think very early question from Anis is, can you please elaborate on the encoder part in the multi-graph GAN? Is that a GNN?
Yeah, that's a GNN. That's a GNN. It uses graph convolutional network, GCN. It's a GCN.
Okay.
Yeah.
All right. The second question from Anil is, do you have the ground truth connectivities, and how did you validate the connectivities predicted by the GNN?
Very good. We do have the ground truth for all of them. So we train. It's exactly, guys, when you have like label data, or when you have the ground truth data. So we do have the ground truth. We split the data into training and testing. During training, we use both ground truth source and target. And during testing stage, we only feed the source and we predict the target, but we still evaluate it against the ground truth. So we use cross-validation for that. But the data set that we have, it has all the observations. Yeah.
Okay, wonderful. I think there's one more, maybe just one more question before we finish here. So what's the measure of connectivity that you use, and are these network compatible with the effective connectivity?
Oh, we didn't use any effective connectivity. That's a very good thing. So we use functional, typical, like just Pearson correlation, PCC based connectivity. We didn't use any dynamic functional connectivity, for example. That's another area that we can dive into once we have if we have the data set or a similar data set. So there are different types of connectivities we can use. Also, like I see that there are a few questions about EEG. So yeah, like it would be so interesting to transform, for example, EEG onto functional somehow using, these kind of networks. So once you guys have the data will guide you into posing the right problem, and you can look at these, methodologies as a starting point, and then you can tailor them depending on the data that you're having. But you can use them for on multiple data. For example, if we have dynamic connectivity, we can try to learn how to predict not only cross resolution, but cross resolution and time at the same simultaneously, which means you take morphological network and you predict a dynamic functional connectome that depends on time. So that's an exciting problem also to solve. But for now, these are still, I would say, foundational preliminary works, and there are so many directions to build on that and take them forward. Yeah.
Okay, great. Maybe just one last question from myself. Okay, so you extracted the brain connectivity from T1 weighted images. So do you still actually use those image information in your network? It seems actually you focus mainly on the connectivity itself, but not much image anymore.
That's a very good question. So yeah, like so far we only use the network. So our starting point for all these models, or these GNN models, are graphs. So we start from a graph, a multigraph, a hypergraph, a time-dependent graph, a single graph. So all of them are graphs. But it would be interesting at some point. I've seen a few works also, like where you combine imaging with graphs. So you try to use, so I'm not sure if it was one of your works, but yeah, so like I've seen, like I remember like bumping into a paper on that. But yeah, so that was actually interesting, combining your imaging biomarkers with the graphs. And it would be also interesting to learn how to predict the image itself and the connectome at the same time. So you generate across Euclidean and non-Euclidean domains at the same time from a single observation. That would be an interesting problem to solve, because at the end, we got that graph from an image. So you can start from the image, infer the graph, and infer multiple images, and then infer multiple graphs. But then we need to make sure that whatever we're inferring or predicting is biologically plausible. And that's the real challenge, I guess, particularly in healthcare and imaging. Yeah.
Yeah, wonderful. Thank you so much.
Thank you.
There are a lot of thank yous in the chat window. You can look at
Yeah, I
yourself.
can see lots of comments here. Sorry. Yeah, like I can take, if I can, let me just screen through maybe. So yeah, I'm happy to, for Neuromatch Academy, happy to collaborate. Just drop me an email. Like you guys have my email, I guess, like it's islam.rekik@gmail.com or i.rekik@imperial.act.uk. I can find it on Google. So yeah, so there's another question. Clinical validity of generating fMRI image morphology given the complementarity of modalities. Yeah, so we have the clinical validity, the cross domain. So basically you validate the functional, we're not generating images, we're generating graphs we're generating functional connectivity graphs from morphological connectivity graphs. So the morphological connectivity graph, that's a very interesting topic. I'm intending to write a review paper on that. We have so many works that show that morphological networks can, in autism, in Alzheimer's, and also regular development, like, a typical development, they are able to identify different types of biomarkers that are in line with functional connectivity biomarkers and diffusion structural connectivity biomarkers. So I believe, like there's a lot of potential in morphological networks in the field of connectomics in general. And it would be interesting to, if we can collect, if we can have a large-scale data set where we compare the biomarkers identified across these different, connectome types, morphological, structural, which diffusion, and functional, that would be interesting. But without even looking at the connectivity, if you look just at the literature of morphological changes in the cortex, for example, or different regions, there is a lot of correlation between changes in morphology, i.e. function, i.e. cognition, in the literature in general, without looking at interactiveness or connectivity. But then at the level of so that's a lower level, like you look just at the image. But then if you look at higher level, which is modeling interactions and connections, that we build on that too. So we assume there should be some changes in, like correlated changes or causal changes between these different facets of the brain. Because if you have atrophy in, for example, if you have atrophy in particular in the hippocampus, so then you might think that atrophy might be correlated to a cognitive impairment in that region too, right? So these facets are kind of intertwined, but we still don't understand how much they are intertwined. And I think we need more connectivity data. We need to have more analysis and processing of the available data sets to kind of explore this. But it's a very interesting question. And I'm very interested in that. I'm just waiting for the data, or like to get the data, or like get started on that. But yeah, so yeah, thank you. And so let me look at the other questions. So I think that's it. We covered the most important ones.
I think that's it. Yeah, you
explainability.
did
There was one about, yeah. So there was one about explainability. Here, explainability for the generative model would be kind of interesting. Explainability for classification might be more interesting, but for generating target from source, it might be interesting in the sense that you're trying to understand how the function is generated from the morphology. So I haven't explored that, but that's a good question to ask. So first we need to ask, is it important to have that explainability when you're generating? So first, like if it's important, then we can dig deeper. But in classification, it is important, to understand what leads to that label of, brain with dementia, healthy brain, etc. So in classification, it's important. In generation or this type of learning tasks, yeah, I need to think about that. Yeah, but good point. Thank you.
All right. I think we are already past 10, so let's just conclude the webinar here. Thank you so much again, Islem, for the wonderful talk.
Thank you. Thank you so much for having me and for inviting me. This was really exciting. Cool. Thank you.
Great.
Wishing you all
see you soon.
Great day.
Yeah.
Thanks.
Another meeting. Yeah. Bye.
Thank you. Bye-bye.
Bye, everyone. Thank you for attending the talk. Thank you.